Hi, we are Group 100. In this project, we grow watermelons. We have two fertilizers. The first one is A, which is currently widely used by farmers. The other is K, which is newly developed. We are comparing the performance of the two fertilizers. As a better fertilizer produces larger watermelons, in this experiment, the parameter we choose to measure is the diameter of watermelon. From the research we've done, it is known that common fertilizers work best at 25 tons per hectare, so we assume this is true for A. However, as K is a new fertilizer, it may not work best at the same level. Therefore, in the project, A is applied at 25 tons per hectare, while K is applied at three different levels, which are 18, 25, and 32 tons per hectare. Here are the hypotheses of our study. The null hypothesis is that the means of the diameters of watermelons under the four treatments are the same. On the other hand, the alternate hypothesis is that the means are not the same. This is our experimental farmland with the dimensions of 500 meters times 500 meters. On the south edge of this farmland, there is a small stream running from east to west. Besides, the farmland is inclined as shown. As the stream may make the soil nearby more fertile and result in bigger watermelons, the experimental site is kept 100 meters away from the stream in order to minimize the effect of it. The site is divided into four regions of same dimensions for the four treatments, and the regions are 20 meters apart from each other. Each region consists of 60 plots. 1/4 of the plots, that is 15 plots, in each region are randomly selected using a random number table for planting watermelons. In each plot, there are 64 plants. The density of the plants is around 1,000 plants per hectare. It is relatively low for better quality of watermelons. This chart shows the levels of treatments. As mentioned, we have four treatments in our study. Each of the treatment contains 15 plots. Each plot contains 64 plants, and each plant is limited to have 2 watermelons. As a result, the sample size of watermelons for each treatment is 15 times 64 times 2, which is 1,920. The same is true for the other three treatments. Then, let's get started to grow watermelons. It is known that the growing period of the watermelon is about 8 months, from March to October. So, in March, we plow to turn over the upper layer of soil. Then we sow the seeds into 3 cm deep holes. There is 1 hole in every 2 meters times 2 meters spacing. 3 seeds are placed in each hole. In the second week, we choose 1 seedling out of 3. The individuals of moderate heights are selected. A week later, we apply the treatments of fertilizers at different concentrations. Watering is generally not required, but if the farmland does not receive rainfalls for a month, we will water the plants. We apply the fertilizers again when veins appear and when the plants flower. In addition, starting from the second month, we apply ambush to control fruit flies and bandlate to control anthracnose. If weeds appear, we will remove them by hoeing. After 4 months, we select the 2 largest watermelons on each plant so as to standardize the number of watermelons per plant. On the 240th day, we record the diameters of the watermelons by direct measurement. We measure the diameters using digital caliper. The diameters of the watermelons are recorded to a data sheet. Dead or deformed watermelons are neglected. To test the hypotheses, we use one-way ANOVA, as we are comparing the means of more than 2 samples, and there is only one factor, which is the treatment of fertilizer. Before performing one-way ANOVA, we need to check the normality and homogeneity of variances of the samples. To check the normality, these 6 steps are used. To check the homogeneity of variances, we use Fmax test. In this case, we assume that the data pass these 2 criteria and proceed to one-way ANOVA. Otherwise, the non-parametric Kruskal-Wallis test should be used. If the result of one-way ANOVA concludes to reject the null hypothesis, this shows that there is a significant difference between the means of the watermelon diameters under the 4 treatments. Then, we carry out Tukey test to identify which mean is significantly different from the others. For example, if the mean for the treatment of fertilizer K at 18 tons per hectare is significantly different from the other 3, and it appears higher, we can conclude that the fertilizer K is better than A with maximum activity at 18 tons per hectare. If the null hypothesis is not rejected, we will repeat the study and work on further hypothesis testing with other levels of K, such as 4, 11, 39, and 46 tons per hectare. We identify several confounding factors, which include rainfalls, pests and weeds, as well as the provider of the seeds. And here are the ways to control the quality with respect to precision and accuracy.